Deep inside you, right this second, there's this incredible microscopic universe just buzzing with activity. And in that universe, tiny little guardians are working around the clock to keep you safe from total chaos. Today, we're going to zoom way in to uncover one of the most important guardians of them all, a secret gatekeeper that literally holds the key to our health at the cellular level.
So let me just start with a question for you. When you think about what protects your body, what comes to mind? Probably your skin, right? Or your immune system? Well, what if I told you that the most powerful protector is a molecule you've probably never even heard of?
Meet glutathione. It's called the master antioxidant for a very good reason. Our bodies make it all the time, and it's basically this tiny little protein that acts as our cell's #1 security guard, repairman, and janitor, all rolled into one.
And trust me, its to-do list is huge. It does everything from cleaning up damaging things called free radicals to repairing cellular damage, building tissue. It even helps our immune system. Honestly, our bodies just wouldn't run without it.
Okay, but to really get why this one molecule is such a big deal, we need to go on a little journey. We're going to zoom right past the organs, past the tissues, and go straight into the heart of a single cell to see this thing in action.
Welcome to the endoplasmic reticulum, or just the ER for short. The best way to think about it is like a super busy factory that's inside every single one of your cells. And its job? Oh, it's only one of the most important jobs there is. Making the proteins that make you, you.
So here's how the assembly line works. Raw proteins show up at the ER's door. Once inside, they have to be folded into these incredibly specific, perfect 3D shapes. It's kind of like cellular origami. If the fold isn't perfect, they're useless. Only the perfectly folded ones get shipped out to do all the important work in your body. But here's the catch. This factory is, well, let's just say it's a little bit picky. For this whole protein folding assembly line to work at all, the environment inside has to be absolutely perfect. And this is where our story starts to get really interesting. Yeah, the ER is a total diva. The chemical balance inside has to be just right. It can't have too much of one chemical charge or too little of another. If the balance is off by even a tiny bit, the entire production line just grinds to a halt. And I'm not just making this up. Researchers from Rockefeller University have the perfect description for it. They call it a Goldilocks-like environment. Not too hot, not too cold. It has to be just right. So how on earth does the cell maintain this perfect Goldilocks state? Well, it all comes down to the two different faces of our hero molecule, glutathione. There's the reduced form, GSH, and the oxidized form, GSSG. The secret is all in the ratio between these two.
And here's the twist. Unlike almost every other part of the cell, this ER factory needs an oxidized environment to work. That means it needs to keep a high ratio of the oxidized GSSG. And it does that by constantly pumping the oxidized GSSG in while kicking the reduced GSH out.
But, that raises a massive question, right? For years, scientists knew this balance was critical, but they had no idea how the cell was doing it. I mean, what was the mechanism? Who was the bouncer at the factory door, making sure everything was just right?
Seriously, the machinery that was running this whole delicate dance was a complete black box. It was one of those fundamental mysteries of cell biology. But a huge breakthrough from those researchers at Rockefeller finally cracked the case.
So allow me to introduce you to the gatekeeper. Its official name is SLC33A1. This is the molecule that stands guard at the door of that ER factory, and it's the one deciding which form of glutathione gets in and which one gets out.
It's what's called a transporter protein, and it sits right in the ER's wall. Its one and only job is to manage the traffic. It pulls in the oxidized GSSG the ER needs, and it pushes out the reduced GSH the ER doesn't want. It is the master regulator of that Goldilocks environment.
Now, this discovery is such a huge deal because it helps us finally understand what happens when things go terribly wrong. What happens when that gatekeeper, that bouncer, just stops doing its job.
Well, it's like a domino effect. If the SLC33A1 gatekeeper messes up, that delicate balance is completely shattered. The factory's environment gets all messed up, which jams the whole protein quality control system. Suddenly, you've got these misfolded junk proteins piling up everywhere. And eventually, all that junk can actually kill the cell.
And this isn't just some abstract cellular problem. As one of the study's authors, Mark God, puts it, this exact process is directly linked to some of our most devastating diseases, like neurodegeneration and cancer.
Finding this gatekeeper gives scientists a real handle on the problem.
And these are the real-world consequences. We now know that mutations in this exact gene cause something called Hupke-Brendel syndrome, which is a really severe neurodevelopmental disorder. And on the flip side, some aggressive lung cancers have actually figured out how to hijack this system, using all that glutathione as a shield to protect themselves.
So you see, by identifying this one single gatekeeper molecule, we haven't just solved a cool biological mystery, we have unlocked a brand new door for thinking about how to treat these diseases.
And this is where it gets really brilliant. Check this out. For diseases caused by a broken gatekeeper, like in neurodegeneration, the goal could be to design drugs that reduce the glutathione overload and bring things back into balance. But for those cancers that rely on the system, we could do the exact opposite. We could design a drug to shut the gatekeeper down on purpose, causing all that toxic stuff to build up and kill the cancer cells from the inside.
It's a pretty incredible thought, isn't it? All of this complexity, from horrible diseases to game-changing cures, all of it hinging on one tiny little gatekeeper managing the flow of one master molecule. It really leaves you with a fascinating question. Could learning how to control these microscopic gateways be the next giant leap for medicine?